like even if I'm not super technical, it will still be like useful to understand. Okay, so let me share. You should enable the sharing for me, otherwise I cannot share.
Web, could you make him
Yeah,
co-host?
give me a second. Uh, uh, yeah, it should work now.
So, ooh. So can you see my screen? Okay, great. So, thank you all for the enthusiasm and for inviting me. As Arini anticipated, I'm gonna talk about food today with a little bit of physics. And yeah, the topic will be to study the face behavior of a famous Italian dish called the cacio e pepe. You can see it here. Can you see my pointer, right?
We can.
And so, in a few words, it's basically pasta with a sauce made of cheese, specifically pecorino cheese, and can be very difficult to like do properly. But as I will try to convince you, there is a scientific way to tackle this difficult task. And again, because you seem all very involved in physics and in technicalities, probably the best way to explain the dish is by using some equations. So let's get some hardcore mathematics here and present you the fundamental equation of cacio e pepe, which of course has involves some pasta. And no, it's not optional to choose like the right shape of pasta because, as you know, in Italian cuisine, the shape has to match with let's say the physical properties of the sauce. And in this case, it's a smooth sauce, so it's very appropriate to use spaghetti, which are long and can really grab a lot of these viscous sauce. But also rigatoni are quite fine. This is a little bit more traditional in this sense. So like pasta, and please buy Italian because like our economy is not thriving lately. So like if you can buy Italian pasta, it's always a better thing. But I guess like you are far away in Asia, so like I will excuse you if you don't find Italian pasta. To other Europeans, I would not excuse them. Then you should use some cheese. Usually you use a pecorino romano, but it's also true that pecorino romano is kind of difficult to find outside of Italy, but also Europe. So you can also use parmesan in case. But if you can, use some pecorino romano. And the idea is to form a sauce out of this cheese. And in order to achieve this sauce, you have to use pasta water. I'm gonna spend a couple of words about pasta water in a couple of seconds, and then of course you have to use pepper because cacio and pepe is a pepper, so you have these two main ingredients. Now, what about the pasta water? So the pasta water is often how can I say it, like underestimated uh, mm, um, ingredient in terms of importance. And as I will try to convince you throughout this talk, the pasta water is very important because in addition to water, you have gelatinized starch. So starch is a big peptide which is released from pasta during the cooking process, and the heat gelatinizes it in the sense that these molecules are usually kind of knotted and you find them in a globular form, and then the heat unfolds them, and these peptides, once unfolded, they tend to stick together and form networks. And this is important for the stability of the sauce, but again, I will postpone more technical details about it later on. So now you take the cheese, you mix it with this pasta water, or let's say water at the moment, so without starch, and you try to obtain like a sauce like this. The problem with the sauce is that once you heat it up, the something happens. So you observe, let's say, a phase transition, which is what we inspired us to study this system, because you see, like you have a homogeneous sauce here, a homogeneous state, and at a certain point you get something like this around the 65 degrees. And so, and this is what happens when you don't perform the recipe well. So what you obtain instead of this like inviting sauce, which would make your nonna happy, you get something like this where the sauce is stringy and separated, unpleasant, and you get an angry nonna. So what is going on here? The idea is that, not the idea, the physics behind it is that heat denatures the proteins. And this image, by the way, was not produced with AI, of course. And so the protein denaturation happens at 65 degrees, more or less. And the microscopic properties of the sauce components change, um, determining at the larger scale this macroscopic phase transition. And in particular, what we have is that the cheese contains two kinds of proteins, the whey proteins and the casein proteins, which are found in the form of micelles. Now, the heat denatures these whey proteins, and also it changes slightly the chemical proteins or the chemical properties of this casein. So why is this important? Well, of course, like the whey proteins, they just in nature, they change their chemical properties. And instead, the casein, which usually stabilizes these stabilizes emulsions, like it's not anymore able to emulsify and stabilize these emulsions. To make things a little bit more clear, if you consider, for example, milk as an emulsion which contains water, which is like made of polar molecules, as you all know, and fat. Fat is usually apolar. You have these long lipid chains, and they don't have anything particularly polar on them. Now, it's also known that whenever you mix something which is polar with something apolar, these things tend to separate. But there we have casein proteins which really are able to bind these two chemical species and keep everything together. Once heated, the casein micelles are not able anymore to perform this job. So in the context of cacio e pepe sauce, what you have is that you go from a state here where the caseins are able to keep the phase homogeneous to something where you get two different phases. And so in the place where I work, people have been working about this phenomenon quite a bit. It's called phase separation. And so our idea was to try to characterize this by using this framework of phase separation. Now, these are the technical slides that I added to the last moment, so I apologize if I'm not very technical and not very clear. In case, please stop me and ask questions if anything is not clear. So, just to before going into the technical part, it's important to say that this phase separation is not like only linked to the culinary world, but it has a important role in describing other kinds of phenomena, like the formation of organelles in cells. So you probably all know that cells contain organelles like well, the nucleus, mitochondria, ribosomes, and all kind of stuff. And all these organelles, they all have a membrane. And well, the membrane is important to keep these compartments separated one from each other. And well, ribosomes produce proteins, so you actually need ribosomes for the whole life cycle of the cell. But there are other things that might happen or might not happen, like an infection, for example. So imagine you have a cell that gets infected by a bacteria or a virus or whatever, and suddenly you need some kind of proteins or molecules in order to face these this infection. And how do you produce them? You need some organelles or like some, let's say, tiny factories that produce these particular proteins that you need. So in order to create quickly these organelles, these compartments, instead of building a membrane, which can be very time consuming, the cell basically tunes its physical parameters like concentration of something or acidity or temperature in such a way that the physical conditions drive the system into separating some kind of components from the rest of the cell. So basically, you get these tiny droplets of proteins which do some specific task. So, like creating these membraneless organelles, it's important for many functions. And in order to do so, you use the physics of phase separation. Again, changing physical parameters in such a way that an organelle is created, which is then able to produce something that is needed from the cell. So this is why phase separation nowadays it's so hyped, because it explains how these compartments are created physically. From a mathematical point of view, we can try to describe a phase separation as the coexistence of two species. Let's call them A and B. And again, you take these A and B and you put them, distribute them randomly on a grid, and you would obtain something like this. So you see this is like a homogeneous phase where more or less everything is homogeneously distributed. And a phase separated state instead would look something like this. So you see you have these red regions and these blue regions. And the idea is, how do you get from this to this, given that at the beginning you don't have any interaction between A and B. And yeah, so the idea is that the first object needed to describe this kind of systems is the mixing entropy. By the way, like I'm receiving messages and there are sounds of all kind. I don't know why. So maybe I can try to fix this in some way. Are you hearing like these notifications and no? Well,
We're not hearing any of that.
okay, then it's only on my computer. Okay, good for you. Then okay, we the first object needed to describe this is this mixing entropy. So the mixing entropy basically tells you that if you randomly distribute A and these two species on um, on um, on a grid, then this mixing entropy is maximized when the mixing is maximum. So you see, you have these two species, A and B, and the entropy is at its maximum when you have a half of one and half of the other, and they are all well mixed. In a certain sense, this reminds us the property of entropy, which if you don't do anything to a system and you let him equilibrate, you would like the most probable configuration that you see is a configuration with the maximum entropy where everything is mixed, right? And in order to get something different from a homogeneous and mixed state is to allow for particles to interact. And in particular, you see like here, if you don't do anything and you mix, so like you allow for particles to swap with the random laws, like you will still always be in a state where everything is quite mixed. Now, what happens if you add some rules for neighboring particles to maybe like to stay close to each other if they are the same type of particle, or they don't like to stay together don't if they don't are not like the same kind of particle. So in this case, you can intuitively see that you will like the energy will tend to like this is similar to what happens in um, in statistical mechanics, where you have two systems which wanna minimize the surfaces between two species, no? Because like in order to minimize these kind of interactions, you will form a bulk where all similar particles are the same, and these volume these in such a way that because the volume scales as the system size, while the surface scales as like a less powerful power law with respect to the system size. And so if you add, so like this was the mixing entropy. And now if you put the interactions, you can say, okay, I can build a free energy of the system, which in addition to the mixing entropy part, you also have something which is which accounts for the interactions between the two species. So if you add this part here, basically the free energy for some particular parameters changes and has two minima here instead of one minima at the center, meaning that if the parameters are tuned at the right point, you will end up a system that either is here or either is here. So effectively separating the system. In other words, like you would get something like this, in such a way that in one region you will have an abundance of a certain species, and in another region you will have an abundance of another species. Ciao, scusa, sono in call. Okay, this was the moment that I told you. So again, this was a more or less very quick and probably not very professional introduction of what's the philosophy behind phase separation, but like let's summarize very quickly. Usually you want to write a free energy where you have a competition between interactions between the species that you are analyzing and something that accounts for the entropy. If the entropy wins, then everything is mixed. If the interactions win, then you have this phase separated state. And again, in the view of a cell that wants to trigger a separation in such a way to obtain a specific organelle to do some biological function, he might tune this parameter here, which is the interaction, or like a local temperature, and then the physics does the rest. So if somebody has any question, because again, this is not exactly my field, this is my first work on phase separation was through Cacio and Pepe, so I guess I might have not been super precise. So please ask questions if something was not clear. Otherwise, I can go back to the less technical part, which is trying to understand what is actually going on in the context of Cacio and Pepe. Now, I anticipated that the starch plays a prominent role in determining the stability of. There is some things in the chat. Oh, Steph, the red spot in the middle of blue just disappeared without traveling again. Ha! Yeah, this is true. So I think in this case, you are not seeing all the time steps. It's just that you see the steps are one. You are seeing like at the 100 at the so every 100 steps. So like these guys here that disappear here in this 100 just went back into this bulk. I think this is why you are seeing pieces that are kind of disappearing. This is flowing inside here. And the one that was here was flowing inside here. I hope this was, are there other questions? Okay. Other questions? Ta, ta, ta. Okay, no, I can continue. Now, what does starch do? But before understanding what's the effect of starch on the sauce, we have to understand what happens to the starch once it's cooked. So when you put pasta in boiling water, actually one of the most important effects of the cooking of the pasta is indeed the starch gelatinization. And this is why pasta has this kind of not gummy, but has this jelly-like consistency. These the starches, which are at low temperatures, are like in a granular form. These also, if you buy powder starch, it's powder. Then you put it in water, and high temperature basically make the granules absorb water. They increase in size until they burst. And once they burst, they are in this free form like these, and they can bind together. And once they bind together, they form these networks which give the jelly-like consistency to the pasta. And also, once you cool them again, they form these networks. Also, you are probably familiar with these videos on YouTube where you put a lot of starch in water, and you form these non-Newtonian fluids where like you poke it very, very, very slowly, and you can penetrate them. And if you do it very quickly, then it works like a solid. So there you are not gelatinizing starch. It's true, you keep it at a low temperature, but also you need a lot of starch to obtain that kind of effect. Here instead, if you gelatinize it, you get something slightly different, but you get this effect with much lower concentrations of starch in water. So I'm talking too much, I know, I'm sorry for that. But like, this is the idea. Starch in water, once cooked, gelatinizes and forms networks, which is super important for many other things. Also, it's a phase transition, by the way. It's super interesting. Like, you can do it at home. It's every time is very fascinating. Now, in order to study the phase behavior of Cacio e Pepe, we are actually using these considerations to avoid using pasta water, which I know it's the traditional thing that every nonna would do, to use this pasta water to emulsify the sauce. But for our purposes, it was kind of complicated to use this because you don't have a full control of the starch content within the pasta. Instead, if you buy like powder starch, what happens to it, like you can really measure how much starch you are using, and then you put it in water, you bring it to a boil, and like you get these, I hope you get a little bit the color of this gel, like it's opaque and it's very viscous. But this is a starch gel. Starch gel allows us to perfectly control the amount of starch in the sauce and allows us to study the physical properties of the sauce. Now, in order to study the sauce in different physical conditions, we decided to build an experimental apparatus, which, by the way, it's very funny because like we are all theoretical physicists, and we built this experimental apparatus from scratch. So this was the original idea, but what we end up with was this. This is a sous vide cooker. I deeply recommend it to cook it in a scientific way. It basically allows you to keep the water at a fixed temperature, and then you have this pot where you put the sauce at the different concentrations of starch, water, cheese ratios, like all the ones that you are interested in. And then you have this other thermometer that allows you to measure the temperature of the sauce within the pot, because there is always like a 5-degree difference between the temperature of the heat bath and the temperature of the sauce. Once you have this sauce, how can I say, this sauce prepared, you will put it you will scoop a little bit on a petri dish, and then take a photo with, again, this very state-of-the-art experimental apparatus, and then you will get something like this. So like anticipating a little bit what I'm gonna say later, you have like a state where more or less the sauce is homogeneous. You will still have some small aggregation, but this is like minor, and in any case, like again, anticipating a little bit, starch will determine whether you end up in this state or in this state. But starch is not a perfect emulsifier. It's not an emulsifier at all. Like technically, emulsifier has like a chemical effect in the sense that it has a polar and an apolar end, and it allows to bind two components without making them form big droplets like those that you saw before. But still, it's not a perfect effect, so you will end up with some small pieces like this. If you don't use a starch above 65 degrees, you get something like this. And I hope this is clear enough to convince you that it's a little bit like the situation that I showed you before in the theoretical introduction, where you have like a phase here, which is basically only water with a little bit of other stuff, and here instead you have everything else, so the proteins, like almost a solid-like phase. It's actually a gel phase, which has some properties like a solid, but it's not exactly a solid. Like for us, it's like two different phases and you can see it. And the good thing for us is that actually you can see it. So what we can do is to segment all these clumps. This was done by my flatmate, who is like really like he's a dragon when it comes to image processing. So like we were able to segment all these small clumps, and then we were able to characterize them by approximating them as an ellipsis. And once you have an ellipsis, you can say, okay, then this ellipsis is characterized by the major axis length. We could have also used the area or other measures. Like here, there are two reasons why we chose to like characterize the system's state by looking at the length. The first reason is that when you do a wrong cacio e pepe, I hope you remember like this photo of cacio e pepe with the strings. So like the stringiness is really a measure of how much the sauce is going south, but also because the phase diagram that we obtained was better. So like very, very honestly, it's like this. But then also with other measures, actually the phase diagrams were kind of decent. But yeah, let's say that we decided to use this measure to characterize the size of a clump. And then, because we are talking about phase transitions, we needed an order parameter in order to characterize the phase diagram. And as an order parameter, we just took the average of all sizes of all clumps inside the sample. So here, for example, you would have taken all the sizes associated to these clumps. You take the average. This number characterizes the state of the sauce at these temperatures, at this starch concentration, at these well, no, these at this temperature, starch concentration, and protein concentration. These are the three variables that we examine, but I'm gonna tell you a little bit more in a second. So here was our first result. So again, we had in order to get a phase diagram, we have to choose two variables to vary, and the first one is of course temperature, because StatMech is always about temperature, and then starch percentage. So starch again was also motivated by the empirical observation from all Italian, also not only Italian nonnas, by everyone who tried to cook a cacio e pepe knows that more or less if you don't use the pasta water, it's very difficult that you will get a smooth sauce. And these like can be quantified by the starch percentage, in the sense that if you have like 100 grams of cheese, in this case we had 100 grams of cheese, 100 grams of water, and then 0.5 grams of starch. So this percentage refers to with respect to the cheese mass. So here it's 1 gram of starch, 2 grams of starch, 3 and 4. And as you see, if you are below this critical concentration of 1%, once the temperature goes up, you end up with this huge clump, which basically spans the whole petri dish, and ends up in this state that we were calling in a very friendly way the mozzarella phase throughout our experiments. But then we decided to leave it on the paper, and people really like it. We like to say that it is a newly discovered state of matter, the mozzarella phase. So this nice name, but not nice to obtain it while you're cooking. And indeed, if you see like if you are at 1% or more, you never end up in this huge clump state, but in a more homogeneous state where maybe you have some kind of clumps. But all these sauces are fine to eat. And so yeah, then here was a little bit of smoothing just to see a little bit better these boundaries. But here is the first result. Like there is a critical value above which the mozzarella phase does not happen at all cooking temperatures, which is, again, you have the sauce, you have the pasta, which was cooking at 100 degrees, you put it together, you want to be here, you don't want to be here. And then, like this, a little bit more physically inspired, what we could do was to vary the water percentage. Like before, we had 100 grams of cheese, 100 grams of water, and then we vary the starch. Here we put ourselves at 1%, because here we don't get to the mozzarella phase, and we vary the water percentage. And as you can see, we observe like a curve where below this, you basically don't have any kind of separation. Yeah, you still have something. But here you have like a bigger clumps, which are not huge, but they are there. But what we really wanted to have is like a curve like this, because it's important from a theoretical point of view. Because we are all theorists. We did a lot of experiments. We said, look, if we are gonna do a paper that is only experiment, this is not very honorable. We have to at least give some model, some equations. So like we were really looking for this. And why are we looking for this? Because okay, this you don't care, because actually, if you go back to the free energy that I described you before, this free energy can give you like an analytical formula for this curve here, and it's a transition curve. Now, here, like I already told you a little bit what these phi's are. In this case, it's a mass fraction of the cheese proteins, because the proteins are what are driving the phase separation. This n is a fitting parameter, and like it tells you a little. So these sides tells you that if a molecule is very big compared to the other, geometrically it will be more likely that two big molecules will find each other with respect to two small molecules. So this is the explanation of this size factor. And this is the interaction strength. And the interaction strength, again, as I told you, is something that cells might want to tune in order to trigger the phase separation or not. And here it has a temperature dependence. Now, if you look at the free energy, the free energy can be used to calculate the chemical potential and the osmotic pressure using these formulas, which you have to believe me are true. And the phase, this transition line is a transition where you go from a mixed state to a phase separated state, and is characterized by like the so-called phase coexistence, like between, like at the line, like it's like the triple point with water. At the triple point, you have a coexistence of all phases. There are some very cool videos on YouTube where you see like that at the same time you have a liquid and water, and liquid and water are the same. So liquid, solid, and the gas, like at the triple point. Here it's a little bit more easy, like you have a line. On the line, you have the coexistence of phases, and the coexistence of phases is characterized by the same osmotic pressure and the same chemical potential of the two phases. So phase 1 and the phase 2. So these formulas here are analytical, because like the free energy is analytical here, and this gives you a set of coupled linear equations which can be solved for the interaction and the size as a function of these lines, the F1, the phi 1, and the phi 2. Just to make, so the phi 1 is this branch here of the line, and the phi 2 is this other branch here, okay? So these values here could be extracted from the experiments, okay? This is why we had what we had here. We have these points, we have them. So we can plug them here in these functions, and we can get, as a function of the temperature, the behavior of the interaction and of the geometric factor. And as you see, actually, the interaction doesn't increase so much. It goes like the increase is very, very small. Instead, the size increases like of an honest factor. It's not huge, but apparently it's enough. And the explanation here is that temperature denatures the proteins, and the proteins unfold. If the proteins unfold, they are bigger. And if they are bigger, they are more likely to find each other and to interact. So this, in a certain sense, tells you that what is happening in the cacio e pepe is not an increase of the interaction strength, but rather it's a geometrical effect. By the way, this might be wrong, because like here, what I gave you, it's this is very funny, you know, this thing about physicists that we tend to overuse the harmonic oscillator to describe everything. Well, we do it because it works. But here, let's say this is the harmonic oscillator of a phase separation. So we are using a harmonic oscillator to fit something that contains a lot of stuff. So this might be wrong. But fun enough, we talked to our supreme boss of the institute, who is one of the most important people working on phase separation in the world, and we asked him, "Would you believe what we are doing here?" He said yes, and that was enough for us. So I see other questions here. Maybe I have to check a little bit. The penguin and the sphere. Yeah, exactly. Or I know it as the cow approximated as a sphere. So if there are three things, A, B, and C, if there be are some phase and C is different, it understand it would be separated, or we would. So like when you put more components, things can be very difficult. So here you see you have two phases because you have two components. If you have three components, you can have three phases. And this is actually what we believe it's the case of this state here. So you see we have a phase where everything is quite homogeneous, a phase where you have like a partial phase separation, and here we have the mozzarella phase. This is a gel-like phase, which might be explained in terms of theory from a system that has three components. Like, for example, you could say, oh, I have proteins, denatured proteins, and water instead of just proteins and water. The only problem is that, like, from a theoretical point of view, this is true. I mean, you can describe these using three components, but you can't do this anymore. In the sense that here, the only thing that we can observe is something like this. And if we had like three phases and three components of the system, you would need here another equation with another variable. And this is the only thing that we can measure. So maybe we could, if we use some microscope, we are gathering founds. So like stay tuned. But for the moment, like this was a little bit too complicated, and also we didn't have that much time. We actually work on more serious stuff on our free time. So like we didn't have time to explore this. But again, stay tuned. I hope you this answered the question. By the way, how much time do I have left?
We're fine with you going as long as you want. Like usually they are hour, 10 minutes, so you've got enough time to do whatever you need to do.
Okay. So Olive Garden is like a vested party. Don't go to Olive Garden. Like I heard it's very bad. But yeah, in any case, I'm gonna go through like I'm almost finished, but I think it's not even that heavy as a talk. So I will say everything I wanna say. So now as a more uh, let's say technically involved part from the culinary point of view, what we have here is that, as I told you, so starch. Here we use a pasta water risottata, which means this is another cooking technique that aims to increase the amount of starch in the pasta water. But like the idea is that even if you increase the amount of starch like substantially, you will always have some clumps. And also you cannot increase it like arbitrarily, because at a certain point it's too much starch and then the sauce is stiff and it's not good. The idea is that this is an imperfect stabilizer, the starch. There might be something better, at least from a chemical point of view. And the protagonist here is this guy. It's trisodium citrate. So this is used a lot in the food industry because it really basically shuts down the aggregation of caseins. And I mean the chemical action is can be it's not difficult to explain, but it's not even so interesting. So just believe me that this is something that really works well, and the people use it in the food industry like all the time. And as you see, like if you don't use enough trisodium citrate, what you have is again a mozzarella phase. This a mozzarella. It's a little bit more strange compared to the case where you are using nothing. It's like it has a weird texture. But then once you have enough trisodium citrate, you see basically everything is super. Look at this. Like there is no sight of any clump. So like Nonna could be happy, or actually no, she's not happy. She's happy for the absence of clumps, but she's not happy because it is definitely not traditional. But the good news here is that it actually the taste it's not that good. Like I as a kid I used to eat these like American cheese that you put in toasts, and you put them in a toast and then in a toaster, and it really has to melt. And if you like, why does it melt and it does not aggregate? Like again, like this is cheese. Like if cheese is above 65 degrees, it should aggregate, and instead it just melts here because it contains like this trisodium citrate. And but the problem is that the taste of the pecorino cheese that you brought from Italy. I live in Germany. You bring it from Italy and you give it to your friends like this. Years and years of centuries of tradition to really optimize the flavor and the notes and every. And then at a certain point, it tastes like an American cheese. So like I don't recommend using trisodium citrate. But it's in any case it's good, and you will never fail Cacio e Pepe using trisodium citrate. You can buy it on Amazon in case. But yeah, like the idea is we forget about trisodium citrate because like people were already pissed at us enough, not so much, but some people yes, for the fact that we were using gelatinized starch instead of years and years of experience. Yeah, these Italians actually don't empathize with us living abroad and don't having infinite access to pecorino, and so we had to find a solution for that. And the idea is that look at here, so like this region here is a region where you are far enough from the critical line where you end up in the mozzarella phase, meaning that if you prepare a sauce with the cheese with this concentration of starch, you should be fine while cooking. And in order to do so, again, no pasta water, but a gelatinized starch with a mass with respect to the cheese mass of 2.5%. In particular, you have 300 grams of pasta, 200 grams of cheese, then you put 5 grams of starch, you gelatinize it, in 50 grams of water, you add 100 grams of water, and then you blend it with the cheese. So you pre-stabilize the sauce in such a way that the network that is formed here really keeps the protein and the fat droplets in place, effectively screening it from the thermal effects of being put in contact with the pasta, which is at a high temperature. So this is a photo of me and some colleagues preparing a cacio e pepe. And this is exactly the blender that we use. So you see, you have the pasta, it's hot, but you have pre-stabilized the sauce. You mix everything together, and you end up with a nice cacio e pepe, which is not separated at all. Then we like to say that our experiments were done on the Petri dish, so like the samples were small, but like phase transition can only be studied in the thermodynamic limit. So we did also big batches, and it works. So here there are some of the well, some authors and some people appearing in the acknowledgements. So if somebody has seen the acknowledgements, this guy, his real name is Filippo De Bortoli, but we acknowledged him as Tetsuya Spipayashi, is its nickname. People were asking on Reddit who is Tetsuya Spipayashi, so keep the secret among us. And then, yeah, okay, this was all the presentation, so I didn't wanna like flex the fact that we ended up on the New York Times, but it's here, and I left it at the end of the day. And this, like, we were very surprised that we ended up here. Also, yeah, they find time to discuss some minor geopolitical issues, but this is of minor importance probably. And here, like at the end, we won this prize. It was super fun in Boston. If you haven't seen the video, it's actually very fun. It's on YouTube. And at a certain point, Daniel, this guy here, says "Ciao, mama," but this is actually true because our moms were there. So this was the squad that came to Boston. Like there is also a grandma, my mom, my dad, and it was very funny. And this said, I hope, well, 50 minutes, it's not so terrible. This is the only author that did not come to Boston. We are still very pissed at him. But yeah, he's he's studying meditate. I think he's in Nepal now meditating. I think. I hope we are gonna see him for Christmas. And thank you for your attention. I hope you reproduce the sauce. Let me know if you do it and if it ends up if it ends up well, because we are still collecting data, and I'm open to questions.
Thank you very much, Ivan. That was a fantastic, fantastic lecture. As always, you guys can raise your hands and then I'll call you out. Abhishek, you can go first. You've been having
Okay.